Our next speaker is from the Translational Genome Research Institute, Dave Engelthaler, who will talk on sequence sputum for rapid drug susceptibility testing. Dave?
Thanks, Barry. Thanks to the organizers for the invitation to come talk to you guys today about some of the work that we've been doing in the TB world. Translational Genomics Research Institute is a nonprofit genome center that really is working on translating new advances in genomics into human health, whether that be public health or clinical medicine or other areas as well, and veterinary medicine too. We've been working in the TB world now for a few years, and thanks to Tim Rodwell and the group at UCSD and now with FIND, we've been able to move a number of things that we've developed closer and closer to the clinical space. So what I'm going to talk a little bit about is amplicon sequencing. We had some great sequencing discussions earlier and fantastic talks in this session that it's going to be hard to live up to. And now I get to be the opening act for Stefan, which is always difficult. And I think I've done this before, but I play the clown well, so you'll look good, Stefan. We've been looking at amplicon sequencing for DST for a few reasons. We've been using amplicon sequencing in a number of arenas to look for antibiotic resistance testing, HAIs, complex environmental samples. It seemed like a very natural approach to try thinking about sputum, the complexity of sputum, and being able to get a DST. So we see the next generation of DST is essentially going from in vitro analysis of isolates to the in situ analysis of sputum. In amplicon sequencing, essentially the way I look at it is enriching for knowledge within the clinical sample. There is information there that we can turn into knowledge about treatment. Amplicon allows us to do that rather quickly and easily. It's essentially a comprehensive approach. We can look at all the targets of interest for us. Now, of course, we don't know all the targets. We keep hearing about more. We just heard about more this afternoon. But they're relatively simple to develop new amplicon targets to put into an ever-growing assay. Combines the sensitivity of PCR, so we know PCR is really a great gold standard for really high sensitivity, with the information power of sequencing. So it can be as sensitive as PCR, but still have all that information within the targets that we amplify up. Again, we can go straight from sputum or other complex samples to be able to look for it because we're enriching. And it has distinct advantages of speed and ease and cost, and I'll address those to some degree. So our initial approach with developing a DST was really looking at the MDR, XDR targets and including more recently PNCA. With the first targets, those first set of targets, we know what the major mechanisms of resistance are, the major mutations, and we just build those into the amplicons to make sure that we can capture those. With PNCA, it's a little different. As has been mentioned, we've got to look at the entirety of the gene, probably a little bit before and after. And then also probably go in and delete out the known targets that don't cause resistance. And so rather than machine learning, we're kind of CDC learning and being able to hopefully grab that information as it comes out. We've already developed new versions of this or new pools of amplicons to add in to target other drugs, including bedaquiline and delamanid and a few others. So amplicon sequencing, the way that we do it is it's a fairly simple approach, and we're looking to simplify it, but the concept is pretty simple as well. Essentially, we're doing two PCR reactions. First, we need to use gene-specific primers that we've built a universal tail onto the end. So we amplify that up. We have our amplicons with these tails on them. We can highly multiplex. We can look at large numbers of targets within an individual run, as well as large numbers of samples, because we can, with that universal tail, put the sample index onto it. So now we can look at tens to hundreds of samples all at once and looking at tens to hundreds of targets if we need to. We sequence this then on an NGS platform. It's quite likely this could work on a number of platforms. We have really developed and validated this to work with the MySeq platform. It's a fantastic platform for this approach, and the new smaller platforms that are coming out by Illumina I think are going to be very useful. We haven't worked with them. We know that there's other sequencing platforms that are out there. They're just not as readily available for us to produce this assay on, but we likely will into the future as well. And then an important piece that I'll talk about here in a second is the automated bioinformatic analysis. We've developed a tool called ASAP, the Amplicon Sequencing Analysis Pipeline, because that's I think the one thing that really prevents NGS or next-gen sequencing from getting into clinical labs is the fear of the data. And so what we've developed, and don't worry about the schematic, what we've essentially developed is a pipeline that essentially standardizes all the sequencing reads that are coming in, aligns them appropriately, interprets them for the mutations that we're looking for, and then produces a very easy to use, easy to read output that could be formatted for surveillance purposes, for kind of like a almost an antibiogram for DST, looking at a large number of samples across all the drugs that are being looked at. But on each individual sample, we can get a clinician report that can be just available with any internet-ready device or right there on the laptop next to the instrument. This provides then information regarding resistance sensitivity overall to the drugs of interest. It tells which codon in which gene has been found. And then on the bottom, we're able to identify what actual SNP was found within each of those genes so that we can go back and better determine what's the cause of resistance in these. And then we also get a readout as to the percentage of the reads, and that really tells us a percentage of the population that's resistant. In this particular case, it's almost 100%, or practically is 100%. But we can look for heteroresistance, and I'll talk about that in a minute as well. We've validated this now across a number of sample sets. The first one in a paper that's in review by Becky Coleman, who's here as well, and if you guys have questions, she's a great one to track down afterwards. We looked at remnant sputum samples from Moldova from Dr. Valeru Curdu. This was really provided by Rodwell Catanzaro with the Global Consortium for TB Diagnostic Development. These were crude sediments that we were able to then just run with those, do the amplification and do the sequencing on, and had really tremendous success. When we compared to the actual phenotype data that was produced for each of these samples previously, we had excellent sensitivity and specificity across the board for most of them. We didn't have enough samples of all the different drug resistance to get great information yet, but for these we certainly did. And you can see really quite good correlation between the amplicon sequencing and the phenotype. And that makes a lot of sense. We are actually just have really strong power to be able to look for those resistance mutations. If they're there, we'll see it in the sample, and these are really well-characterized mutations that we know to cause resistance. So it makes sense that we had really strong correlation. I think these numbers will start to change as we're adding in drugs that don't have as quite a good information on the mutations that we want to look for. But we have, as I mentioned, we have done some PZA resistance. This is actually fairly new data, samples provided by Rodwell again, coming from India, Moldova, and Philippines as part of their previous study, of which they had already characterized. They were totally blinded to us. We ran the Amplicon assay for PNCA, and we were able to, out of 35 samples, we had 100% sensitivity and specificity. 28 of those were resistant, 7 were sensitive or fully susceptible to PZA. But this is also a very small sample set, and we probably lucked out like crazy. So if you're going to work with PNCA, work with this sample set, and then it'll look really good. We know that as we start to push this out, it's going to be messy, but we can actually just train the bioinformatic tool to just ignore or include SNPs as we learn more information about these. And of those 28, 27 had differing SNP mutations, so that it clearly wasn't the same mutation. These weren't clonal samples that we were looking at. They were just different mutations. They all happened to confer resistance. So that's pretty much a brief overview of next-gen RDST. We can examine the well-characterized SNPs or any SNP within a target amplicon or any indel as well. The cost, we've been working with FIND to help identify. This has got to be the target. We want to get down close to $15 per sample. It really depends on the number of samples that you run through sequencing. If you run 100 samples, that's going to be or if you run 50 samples at a batch, that's going to be a cost of about $30 total in reagent, of course, then there's machine time and operator time and all of that. But it's using the MySeq and hopefully some of the newer tools, it's becoming fairly routine process to do this. It's not really complex library prep, and we're really trying to simplify that over the next year. The time, currently, we're taking about 56 hours because we're doing fairly long sequencing. Our target is to shrink this down to less than 24 hours from sample to answer. And then the detection limit for looking for minor populations, we can actually look down to about 5% heteroresistance within samples, or look for 5% of the reads, which pretty closely is equivalent to 5% of the overall population within the sample to detect heteroresistance, and I'll talk about that more here in a second. And again, this is currently being optimized then for speed and for cost, working with Find and working with others. We're going to try to push this into a single PCR, an easier prep, very routine, hopefully can be automated as much as possible, use shorter reads so we have shorter sequencing time. And then, as I mentioned, automate both the prep side as well as the analysis side. And we've already done a lot on the bioinformatic analysis so that you never have to touch the data. But wait, there's more. There's a lot of power behind doing amplicon sequencing. We can not only get, I think, a really good profile on the DST, or at least a genotypic profile, we may now be able to use the power of NGS to detect resistant markers before actual resistance occurs. Now, this is likely going to show up as, if we're detecting it at very low levels, that's going to be discordant with the phenotype results. But that just is, we think in many cases, that's because it's at such low level that it's not showing up phenotypically. But does that mean that we can detect pre-resistance? And I think that this is a concept that we can start to think about that we really couldn't think about before. We didn't have the tools. And so I think with heteroresistance detection, this is really using a different twist on at least how we analyze the data to be able to find very low-level resistance, and I'll describe that more. So if we define heteroresistance as the presence of mixtures of susceptible and resistant phenotypes in the same population, and that these may come from superinfections of multiple strains with differing phenotypes, or it may come from evolution of the original infecting strain into multiple lineages in situ. Either way, you could end up with a minor population that's resistant, and that may not be detectable. And so now we can identify those very low-level resistant mixtures which don't show up phenotypically resistant yet. And so the question then, is this important? So phenotype testing has been considered theoretically to essentially be able to identify 1% heteroresistance. Now, the data that we've seen, that's probably somewhere between 1 and 10%, depending on the sample. But if you look at one resistant cell out of 100, we know that that's important, that you can select for that, and it may start to take over the population, and therefore we want to know if it's heteroresistant so that we're not treating with an inappropriate drug. But what about 1 out of 1,000? Well, we certainly can't do this with phenotype, and we can't do this with other sequencing methodologies. However, I think we can with next-generation amplicon sequencing. So 1 out of 1,000, or even 1 out of 10,000. It seems like maybe you probably can't even see the red dot on there anymore, but there it is, and it's still there, and it's still going to be selected for if it's alive and you treat with the wrong drug. Is it making you guys dizzy? I was staring at this on my computer. So what can we do? Well, we came up with a methodology a couple years ago called single molecule overlapping reads, or SMORE, where essentially it's amplicon sequencing, but our goal here is to have overlapping reads on every molecule so that we get double confirmation of each of the loci of interest. And the reason for doing this is that we want to get way below the sequencing error. Heck yeah, we can sequence the heck out of something, get 10,000 or 100,000 X coverage, but if you have 1% sequencing error, you can't trust anything at 1% or lower. So we want to get past that. If we're looking at one read, the average we would see with Illumina would be about 1%. Many locations it's going to be well below 1%. Some locations we see sequencing error well above 1%. So we say about an average of that. But if we have double coverage on the loci of interest, we know that we can push that sequencing error way down to about 0.01% or lower. And so everything above that we can have confidence in. So looking back at some of the initial samples that I talked about from Moldova, there was a number of these XDR samples that had evidence of mixtures in them. And I know you guys probably can't read that. I'm not going to go through all this data. I just want to point out a couple samples within these mixtures, and this data has been published. Looking at a couple of the DST-resistant bugs, they clearly, for INH, they clearly had the CAT G315 mutation at different levels. One of them, it was a dominant. 61% of the reads were the resistant mutation, and the other is a lower level, 13%, but clearly enough for it to be phenotypically resistant, and that's the way that it showed up. But a couple other samples were DST sensitive, and we absolutely are confident that it has very low level resistance population, that one at 1.2%, another at 0.33%. And part of the reason that our confidence is that these are very high coverage, and these are these SMORE reads, these double coverage reads at 10,000x or 25,000x double coverage. We're absolutely getting good data for these samples. And so then the question is, does this represent a pre-resistant state? We don't know. We haven't gone back and treated necessarily these patients or these isolates, or the isolates that would have come out of these, to see if we could grow that up to make it resistant. But my guess is that certainly theoretically we can, and likely we could. Another example, looking at rifampicin resistance, these were DST resistant. This one here at 22% coverage, and so comes up both genotypically and phenotypically, as that would make sense. But another one that was DST sensitive at the same loci, 0.45% resistant. And these are, again, very low level, and the question is this considered pre-resistant? And I think these are questions we can ask now and start to explore, because we have the tools to follow them along. And we also have partners that actually can do some nice modeling. Some other samples that we looked at, we've applied this to isolates as well. These are isolates that originated from Rob and have been worked with John Metcalf at UCSF and applying the same SMORE tool. This heat map is just showing percentage of susceptibility, and black is 100% resistance, white is 100% susceptible. So any other color that you see there is some type of mixture that we're detecting with SMORE. And you see there's a number of them that are beige or yellow. These are very low level resistant samples, and so we're able to track this even as we pull these out of culture. I should have another, but wait, there's more, because there's really more interesting things going on here other than just these mixtures that are occurring that we may not have been seeing before. We're also seeing that a mixture isn't just a mixture. It can be a lot more complex than that. And so looking at a couple of those samples from that last slide, for instance, this sample that shows 100% resistance at gyrase, okay, good, it's resistant. We know that it's resistant. That makes sense. We're not going to use fluoroquinolones. But if you go in and look at the actual reads, we see that in this particular sample, it's essentially 50-50 split between two different mutations, both at the 94 codon within gyrase. And so we have almost a complete split of the resistance that's occurring. So it's two lineages that are resistant. Are they different? Were they different original specimens, a superinfection, if you will, or are these both grown up during an infection? And so we can start to hypothesize, models that would lead to something like this. Here we're looking at a proposed pathway that you would have an infection with a strain that had previous concurrent selection together. You likely had a treatment, two mutations occurring similar time early on with fluoroquinolones, and you selected for these, and they became the dominant and the only strains present. But if you look at a different sample, we can look at this sample that was 10% resistant by gyrase and actually included a number of different loci. And in here, it's 90% wild type, and you think, well, 10% resistant, well, that's got to be phenotypically resistant. But in fact, it's not just 10% resistant. It is resistant at we have four different lineages occurring within this sample. And they're all occurring at about 2 to 3%. So that's a pretty interesting finding that we never would have seen before if we hadn't been looking at this data. And you can imagine that the pathway was that there was recent concurrent selection occurring at the same time, same rate of mutation selection happening, that's starting to grow these up and becoming an important part of the overall population. So there's many of these examples that we have, and they even get more complex and I won't even go through this one, 97% resistant, but all sorts of different resistance and probably different models, ways that this could happen. But it's a much more complex infection going on within these patients than I think we had previously identified. And I think because we were going to keep this shorter, I clipped out the last few slides, so I don't have them, but I want to thank all of the collaborators that we have done samples with, we're currently working with, or that we're about to. We have some samples in-house from a number of folks, and a lot of these folks are in the room, have helped us shape our thinking, have helped us develop the tools that we're looking at, and I think are going to help us answer some more of these questions. Most importantly, I have to point out down at FIND and UCSD, both Tim and Becky Coleman. Becky was my postdoc for a couple of years, has recently gone over to work for Tim, and we're still interacting with her, and she's been the brains behind a lot of this. And with that, thank you.
Thank you for a great talk right here. If you are able to detect pre-resistance isolated from a patient, what kind of recommendation would you give to the treating physician in terms of treatment regimen composition for a particular drug, especially a new drug?
Well, I like the hypothetical because it started with if, and so we don't know that that's what we're finding, but that's the question we can ask. Are we finding that? And we're going to have to prove that both in vitro as well as then with patients and follow, do these patients actually select for this in blinded studies? If they do, and if they become selected for, then that could become clinically useful information. Until that time that we've proven that, I don't know how useful that is for the clinician at this point.
Yeah, what polymerase are you using to do your initial amplification? And is polymerase error a potential reason for having to go to bidirectional sequencing?
Yeah, so the question about polymerase and polymerase error. So we're using a high fidelity Taq polymerase because of the potential for that, but polymerase error is so much lower than sequencing error that our bigger concern was to get past sequencing error. So polymerase, our PCR error, is going to be about 10 to the 6. High fidelity Taq, 10 to the -6, 10 to the -7. So that's well below what we're looking for. So we don't think that is going to come into effect.
One more question, because we're getting the...
Devil is in details, and I'd like to ask you a technical question.
Yeah.
So how big are your amplicons, and how long are your reads?
So most of the amplicons are, I believe, about 270 to 370. Is that right, Becky? Yeah.
And how long are your reads?
So the reads are going to be as long as the amplicon, we'll have the universal tail on there, so it's right around 300, 350 is total, because and I didn't throw any reads.
Usually, the reason why I'm asking, nobody is usually doing more than 100.
Yeah, so the good question is, right now we're using the 2x300 kits with the MySeq platform, so we get reads 300 both ways. We can get double coverage on most, but we don't need that necessarily for the DST. We only need that if we want really low or high confidence and very low minor populations. If we're going down to 5%, we can use single reads, or we could use shorter reads, and that's what our plan is, to drop that down to 150, 75, maybe even lower, using some cool tricks that we're talking to Illumina about right now.
Thanks.
Thank you.